Hello dear students, welcome to the second session of the chapter Molecular Basis of Inheritance. In this session, we are going to study four important experiments based on the search of genetic material and the property of genetic material. Okay, we can describe each and every experiment one by one. The first experiment is the transformation experiment conducted by Frederick Griffith in the year 1928. The organisms used by him were mice and a bacterium. The bacterium is called Streptococcus pneumonia. It is a pneumonia-causing bacterium. Two strains of bacteria were used. One is a rough strain or R strain. Another one is a S strain or a smooth strain. So, what is the experimental mechanism? The step one, the mice were injected with the live specimen of R strain of Streptococcus pneumonia. But after some time, pneumonia never occurred, no pneumonia. And due to the absence of pneumonia, all mice survived. Hence, the live R was considered to be non-virulent. Non-virulent means unable to cause the disease. So, inside the body of the mice, he identified the presence of live R. That means there is no transformation of a live R. The second step, he injected live S into the body of the mice, and all the mice were died due to the attack of pneumonia. Hence, the live S was considered to be virulent. Virulent means capable of causing the disease. Inside the dead body of the mice, he identified the presence of live S. So here also there is no transformation. When he injected live R, ultimately the live R will be there inside the inside the mice body. When he injected the live S, after the experiment, live S was found to be inside the dead mice. So there is no transformation in the first step and the second step. Then the third step, he heat killed S. What is the difference between R variety and S variety is that the smooth strain has a mucopolysaccharide covering envelope. That is why the colony is supposed to be shiny or smooth. So he treated the S variety in hot water so that the covering will be lost, and that kind of variety is called heat killed S. When he injected heat killed S into mice also, no pneumonia, all mice survived. Heat killed S was supposed to be non-virulent, unable to cause a disease. So inside the body of the mice, he identified the presence of dead bodies of S. So here also no transformation. So when live R was injected, no transformation. When live S was injected, death occurred but no transformation. When heat killed S was injected, in that case also there is no transformation. In the last step, he injected a mixture of live R with the heat killed S into the body of the mice. The live R was supposed to be non-virulent in the first step. The heat killed S was also proved to be non-virulent in the third step. But when they injected together, pneumonia occurred, all mice died, and inside the dead body of the mice, he identified the presence of live S. That means there is a transformation. He injected only live R and heat killed S, but here, due to the influence of heat killed S, something from the heat killed S, the live R got transformed into live S. Live R is a non-virulent variety, live is a virulent variety. So there is a transformation of non-virulent live R into virulent live S. So here, the transformation of R strain into S strain is due to the transfer of genetic material from heat killed S. Okay, so there is a presence of genetic material is proved by the transformation experiment conducted by Frederick Griffith. Okay. Now, the second experiment is identification of the biochemistry of the transforming principle proposed by Griffith. And this experiment was conducted by Oswald Avery, Colin MacLeod, and Maclean McCarthy in the years 1933 to 44. Here also the same procedure was done. They used the organisms mice and bacterium, Streptococcus pneumonia, R strain, rough strain, and smooth strain, S strain. As per Griffith experiment, some chemical from the heat killed S transformed livar into livus, isn't it? So they isolate the chemicals from the heat-killed yes and injected one by one. The first step, the livar treated with the proteins of the heat-killed yes. Livar are mixed with the proteins of the heat-killed yes and conducted the experiment. But there is no transformation. That means the transforming principle cannot be protein of the heat-killed yes. Then the second step, they injected livar with RNA taken from the heat-killed yes. In that case also there is no transformation. That means RNA of the heat-killed yes cannot be acting as the transforming principle. In the third step, they used livar treated with the DNA of the heat-killed yes. So in that case, the livar got transformed into livus. Okay, the non-virulent livar got transformed into livus in the presence of DNA molecule of the heat-killed yes. So the biochemistry of transforming principle was identified as DNA. Okay, the same experiment was conducted in another manner. So heat-killed yes containing the transforming principle, and the heat-killed yes contain protein molecules, RNA molecules, and DNA molecules. The first step, they isolated these three chemicals and injected each one. The second step, they used protein digesting enzymes. First step, when they are using protein digesting enzymes, the proteins are removed, RNA and DNA will be there. So there is transformation. In the second step, they used RNase enzyme to remove the RNA so that protein and DNA will be there. Again, a transformation occurred. At the last step, they used DNA digesting enzyme, DNases, then so that protein and RNA will be there. So there is no transformation. That means only when transformation takes place, only when DNA molecules are present. That is why the DNA is considered to be the transforming principle. Hence it was confirmed that transforming principle cannot be protein, cannot be RNA. Hence it was confirmed that the transforming principle cannot be protein, cannot be RNA, but that will be DNA. The next prime point is regarding the confirmation experiment for DNA is the genetic material. That experiment is called a bacteriophage infection experiment conducted by Alfred Hershey and Martha Chase in the year 1952. They used a different type of experiment. The organisms used were E. coli and a bacteriophage. Bacteriophage means a virus that infects bacteria. They used the cultures of viruses. The virus cultured in a medium containing radioactive phosphorus, P32, and a medium containing radioactive sulfur, S35. So, in the case of a virus, a bacteriophage virus, you can see its body consists of two types of structures. There is a protein coat. The protein coat of the virus is called a capsid. This is called a capsid. The capsid contains sulfur. And inside the capsid there will be the genetic material called DNA. The DNA contains phosphorus. This is DNA. DNA contains phosphorus. So when they used radioactive sulfur, the protein coat of the virus became radioactive, and when they used radioactive phosphorus, the DNA of the virus become radioactive. That is a condition. The viruses cultured in the medium containing radioactive phosphorus contained radioactive DNA but not radioactive protein, because phosphorus is present only in DNA. Viruses cultured in a medium containing radioactive sulfur containing radioactive protein coat but not radioactive DNA, because sulfur is present in the protein coat called capsid. So there are three stages in this experimental mechanism. One is infection, other one is blending, third one is centrifugation. When the virus attacks E. coli, it will insert the DNA into it. The protein coat remains outside. Okay. So the entry of viral DNA into the E. coli is called infection. The first step, E. coli was infected with the virus containing radioactive phosphorus. The radioactive DNA is inserted into the E. coli and the non-radioactive protein coat remained outside. Then, in the next stage, E. coli was infected with virus containing radioactive sulfur. So that the non-radioactive DNA is inserted inside the E. coli and the radioactive protein coat remain outside. Okay. So whatever may be the case, the protein coat will remain outside, only the DNA enters. When radioactive phosphorus is used, radioactive DNA enters. When radioactive sulfur is used, non-radioactive DNA enters. The entry of DNA is called infection. Then we are separating the E. coli and protein coat by a process called blending. The E. coli infected with virus containing radioactive phosphorus was agitated using a blender. The non-radioactive protein coat is detached. Then E. coli infected with the virus containing radioactive sulfur was also agitated by using a blender. Now the radioactive protein coat get detached. When we are using phosphorus, the non-radioactive protein got detached. When in the experiment using radioactive sulfur, the radioactive protein got detached. Anyway, detachment of protein coat from the E. coli is brought about by the second step called blending. Then the last step is called separation by a process of centrifugation. E. coli infected with virus containing radioactive phosphorus was centrifuged. You know that the E. coli is heavier than the protein coat, so that when we are centrifuged, the sediment become the heavier part and the supernatant become the lighter part. So in the first case, the supernatant is a non-radioactive protein coat, and the sediment have the radioactivity. E. coli have the radioactivity. When E. coli infected with the sulfur, radioactive sulfur was centrifuged, the supernatant contained radioactive protein coat and the sediment contained E. coli having no radioactivity. Understood? So this is the centrifugation tube. Here comes the sediment. Here comes the supernatant. Okay. When radioactive phosphorus is used, the E. coli has radioactivity. When radioactive sulfur is used, the protein coat was radioactive, the supernatant was radioactive. So bacteria, when infected with the viruses having radioactive DNA, were radioactive. Bacteria, when infected with viruses having radioactive protein, were not radioactive. So radioactivity property can be induced in the E. coli only through the agency of DNA, not through the agency of protein. That is why the DNA is considered to be the genetic material which is passed from virus to bacteria. It's a very important experiment. You have to study all the details. So two radioactive chemicals are used, radioactive phosphorus for making the DNA radioactive and radioactive sulfur for making the capsid radioactive. Result is that when sulfur is used, that means when the protein coat is made radioactive, the E. coli get no radioactivity. When the DNA is made radioactive by using radioactive phosphorus, the E. coli get radioactivity. That means the genetic material can be DNA. Okay. Now there is one more experiment in this chapter describing the semi-conservative mechanism of DNA replication. It is an experimental proof for semi-conservative replication described by Matthew Messelson and Franklin Stahl in the year 1958. The organism used is E. coli bacterium, and two types of culture media are used. In one, ammonium chloride with a heavy isotope of nitrogen N15, in another ammonium chloride solution with a light isotope of nitrogen N40. Initially, E. coli bacteria were cultured in a medium containing heavier isotope of nitrogen for many generations. All the newly synthesized DNA incorporated heavy nitrogen, and the DNA can be isolated by using density-dependent centrifugation in cesium chloride so that they made heavy DNA. Normally, the DNA is N14, that is called a light DNA, but they converted that light DNA into heavy DNA. That means in the heavy DNA, N15 will be there. So here will be N15, here also N15. Both strands were having heavy nitrogen. That is called N15 and 15. Then they transferred the cells into a medium containing normal ammonium chloride solution with light isotope of nitrogen. They took samples at various definite time intervals as the cells multiplied and extracted the DNA, and then centrifugation takes place. The DNA was extracted after one generation, that means after 20 minutes. Then they are centrifuged. They find that all the DNA molecules contain one strand heavy and one strand light. That means N15, N14. That is called hybrid DNA. All the DNA is the hybrid type. That means one strand with the heavy nitrogen and the other strand with the light nitrogen. And this is possible only by a semi-conservative mechanism. That means the parental DNA, heavy N15, N15 is isolated, and new strands synthesized will be of N14, that is light type. So when one strand is heavy, another strand is light, the DNA is called hybrid. Then after 40 minutes, the second generation is isolated and centrifuged. 50% of DNA were hybrid. That means N15, N14, and the remaining 15% will be light type. That means the mother DNA has one strand N15, one strand N14. From N15 strand, an N14 strand is added. That means that become hybrid. To the N14 strand, another N14 strand is added, so that become light. That is why 50% is hybrid and 50% is light. So the result shows exactly the pattern of semi-conservative DNA replication. What is semi-conservative? Out of the two strands in the DNA, one strand will be maternal, that is taken from the mother DNA, and the other strand will be newly synthesized daughter strand. And this process is called semi-conservative DNA replication. And the mechanism of semi-conservative DNA replication was very well explained by the experiments conducted by Messelson and Stoll. The similar experiment was conducted using radioactive thymidine by Taylor in the year 1958 in a plant called Vicia faba. So both these experiments proved that the DNA replication is by a semi-conservative mechanism. So these are the 4 experiments described in the chapter Molecular Basis of Inheritance. All the 4 experiments are very important for your examination. Okay, thank you.